Hello, I'm Ron Vale, and I'd like to welcome everyone to Life Science Across the Globe, a Sister Institute seminar series. This series is freely available to anyone in the world, and more information on this series can be found on this slide. And previous talks can be found on our website, lifescienceacrosstheglobe.org. So far, we've heard from Janelia Research Campus, Cold Spring Harbor Laboratory, the University of Buenos Aires in Argentina, the Laboratory of Molecular Biology in Cambridge, and today we travel across the English Channel to Germany and welcome our sister institute, the European Molecular Biology Lab. And today I'd like to introduce Anna Fruzzi, who is the head of the Developmental Biology Unit, and she also serves as director of the EMBL, International Center for Advanced Training. Anne, welcome to the series, and we look forward to your talks.
Thank you very much, Ron. It's a pleasure to be here, and I'd like to take a few seconds to introduce the EMBL. The European Molecular Biology Laboratory is one of the world's leading research institutions, EMBL's flagship laboratory for research in the life sciences. We're an intergovernmental organization, the equivalent and counterpart of the European Nuclear Physics Organization, CERN, but for the life sciences. EMBL was established in 1974, and our role is to promote molecular biology across Europe and to act as a crucible for emerging scientific talent. A large part of our work is training Europe's leading early-stage molecular biologists, giving them everything it takes to launch their independent scientific career. We keep in close contact with our huge network of alumni, many of whom go on to leading positions worldwide. EMBL has 5 key missions. First and foremost, conducting excellent fundamental research in molecular biology and offering a huge palette of training to graduate students and postdocs, as well as professional development for scientists. But we also serve a community of millions of life scientists worldwide by providing a diversity of high-end scientific services, from imaging to structural biology, as well as the world's most comprehensive range of freely available molecular data resources. Another mission is to develop new tools to push the boundaries of our knowledge and to engage in technology transfer in industry, maintaining close relationship with specialist companies. We're able to keep the most of the advances that come as a result of EMBL research. Our final mission is to build bridges across European life science research and to support collaboration within Europe and beyond. Collaboration and coming together is vital. And here I'd like to express thanks on behalf of EMBL for HHMI's work in making these sessions possible. 2020 has shown us the pressing importance of communicating our work and how essential it is to maintain both public interest and public trust in science. So having given you an idea of EMBL in a nutshell, I'd now like to introduce Nassos Typas. Nassos is a senior scientist at EMBL Heidelberg, where he's been running his group in the genome biology unit since 2011. His group combines automated platforms and genome-wide approaches with molecular mechanism to study bacterial cellular networks, how bacteria interact with the environment, the host, and with each other. Key interests of his lab are antibiotics, bacterial pathogenesis, and the human gut microbiome. Nassos is a trained biochemist, geneticist, and systems biologist. He did his undergraduate studies at the Aristotle University of Thessaloniki in Greece, his PhD at the Free University of Berlin with Regine Henge, and his postdoctoral research at University of California, San Francisco with Carol Gross. Nassos has received a number of awards and is a member of the European Academy of Microbiologists. To you, Nassos.
All right. Thank you, Anne, for the nice introduction. Good morning, good afternoon, and good evening to everybody. Taking over from where Anne left it, EMBL has been a beacon for molecular biology research since its foundation. This research is usually performed across scales, going from molecules down to an atomic resolution of those molecules up to the organism level and back. In the future, and as part of our new program, which we hope our member states will support, we want to go one step further and ask and look into those organisms within their complex and dynamic environments. And that can be done at a population level, at a community level, or within the whole ecosystem. So now the questions that arise there, and one can ask, is that how molecules and organisms form complex and dynamic ecosystems, how do they respond and they adapt to the different environments. Ultimately, what we want to do is unravel molecular mechanisms that explain life in context. When talking about ecosystems in this planet, it's good to remember that we live in a planet of bacteria. This is something the famous evolutionary biologist and paleontologist Stephen Jay Gould acknowledged in a provocative and inspirational article more than 2 decades ago, which if you haven't read, I would urge you to do so. So bacteria have been evolving for billions of years in this planet, long before there was oxygen on it, and are able to proliferate in any environment, no matter how harsh this seems, often coexisting in communities like the ones that we see in our gut. These dynamic environments have shaped microbial evolution, but the opposite is also true. Microbes themselves have shaped our planet. Current estimates are that there are more than 10 in the 12 microbial species living in our planet, and more than 10 in the 30 microbial cells. Within this humongous diversity lies an opportunity to identify new functions, some of these which will enable in the future both health and biotech applications. Most microbes live in communities, how these communities forms, what are the characteristics, what maintains their stability, drives their outputs, how they interact with the surroundings are key questions for the future. Here at EMBL, several of us use the human gut microbiome as a prime example to study microbial ecosystems and start answering some of those questions. So in my lab, what we do is that we develop systematic quantitative approaches and combine them with molecular mechanism to look into different aspects of microbial physiology. So we're particularly interested in the diversity of functions and interactions microbes have, and we largely operate on those 5 themes that you see on the slide. For the purpose of the talk, I want to focus on the work we do on the gut microbiome so I can give you some idea of what questions we can ask and answer. So the human microbiome has been a booming field for the last 10 or 15 years, largely propelled by data-driven science. I'm sure that some of those numbers you have heard before, but what I want to say is that this data-driven research has provided impressive insight into the diversity and complexity microbial communities can have in nature. So by now we know that our body carries at least as many bacterial cells as human cells, have 100 times more bacterial genes in or on our body than we have human genes, and most of these bacteria actually reside in the gut. And although individuals may retain the same gut microbiota throughout most of their adulthood life, the microbiome between 2 individuals can actually differ up to 80 or 90%. This is in stark contrast with our human genome, which is 99.9% the same between 2 individuals. Now, over the years, the composition of the gut microbiota has been associated with many diseases and dysbiotic states, initially in the gut, but throughout the years, throughout the entire human body. The issue with association studies is that it's difficult to infer causality and mechanisms without experimental systems. And these have been lacking from the field for many years. And to tackle this, we teamed up actually with the labs of Per Borg, Kieran Patil, and Georg Zeller at EMBL, and we tried to assemble a representative strain collection of the most abundant and prevalent microbes found in the human gut. So what you see here in the left, you see microbes at the genus level that all of us carry in our gut, ranked in terms of the median abundance they have across individuals. And here on the right, you see how they break down in species diversity. Every time you see a gray box, that means that as a species, we have at least 1 strain. In most cases, we have several strains out of it that we can cultivate. And then we have a large collection that encompasses more than 100 species nowadays and several hundreds of strains, which you can use automated experimental pipelines to cultivate, to assess both the characteristics, the interactions, and how they form complex communities. So using this resource, we have answered quite a few questions here. I want to focus on 2 different facets of the same question, and that is how medication affects the growth of gut microbes. This is, again, work in collaboration with Per Borg and Kieran Patil, spearheaded by Lisa Meyer, an ex-postdoc in the lab who has moved now to set up her own lab, started by Michele Brutano, and all the analysis done by Michael Kuhn. So as I mentioned before, individuals differ dramatically in the microbiome composition. So as larger microbiome cohort data became available, research could ask what parameters can explain inter-individual differences. Here I'm showing you one of the first studies from a large Flemish cohort. And as you see, medication is at the very top, explaining inter-individual differences in terms of microbiome composition. Similar results have come out from larger cohorts through the years. And I think the common theme is that environment rather than human genetics define the microbiome composition of individuals. Medication is always one of the strongest parameters contributing into changes between individuals. Now, the second part is that in recent years, it has also become evident that it's not only antibacterials, might be also drugs that they're not bona fide antibiotics that may affect microbiome composition. The poster child of that era is metformin, the leading drug for type 2 diabetes. And there are also reports about proton pump inhibitors, NSAIDs, and atypical antipsychotics changing the microbiome composition. Yet, in all those studies, it's unclear whether those effects are direct and what is the extent of those effects. So this is exactly what we try to address. So having this resource, we could go and test directly the effect of drugs on microbes, what is the extent, are they direct, and whether down the road they have any relevance. Is it part of the side effect of the drug or part of the mode of action? So how did we do that? So we took the Prestwick library, which has 1,200 FDA-approved drugs. The pie here is to make the point that about 20% of them are anti-infectives, but the rest of it, so the rest 80%, are drugs that have cellular targets in human cells, and we turned that as human-targeted drugs. In order to do that, so we took those 1,200 drugs and we profiled them across 40 different strains, representing 38 different species, using automated approaches under controlled environment conditions. To jump directly on the results, what we see is quite astonishing. Actually, we see that the impact of drugs on the human gut is extensive, and that is not only for antibiotics, but actually also for non-antibiotics. We see that at least a quarter of them impact the growth of one gut bacteria. To give you the data for that, this is a little bit of a complex slide, but I'll walk you through. So what you can see on the y-axis the 40 different species that we have on the screen. On the x-axis, the 1,200 different drugs we have, and they're grouped in three different categories. One is antibacterial drugs with green, human-targeted drugs are orange, and other anti-infectives, drugs that would target parasite, fungi, or viruses, are with purple. Every time you see a colored stripe in this plot, that means that this particular drug has inhibited the growth of this microbe. So starting from the antibacterials and using them for the moment as control, we'll come back to them. What you can see is that many of them have broad spectrum activity, so they actually inhibit the growth of almost every microbe in our screen. What gets more interesting is that when we go to human-targeted drugs, well, we see that actually about a quarter of them, so more than 200 drugs, inhibit the growth of at least one microbe. Some of them act across the spectrum, almost like antibiotics, so they inhibit the growth of everybody, but the majority of them actually have quite species-specific pattern. The other anti-infectives act in between the two other groups. Now, what I want to make as a point is that this estimate, if anything, is an underestimate. And the reason I'm saying that is because the drug concentration we've used in the screen is actually on average much lower from what we expect the drug to be in the gut of individuals. We also see that the drugs that are non-hits in our screen are actually taken at higher doses by individuals. And to exemplify that, metformin, our positive control, is a non-hit in our screen. The reason for that is that we screen at a concentration that is 30 to 300-fold lower than what is found in the colon of people that take metformin. When we increase the concentration, metformin directly inhibits the growth of microbes that they're seen missing from the individuals getting metformin. We also use very stringent cutoffs to call hits. We don't want to have any false positives, but this always comes as an expense for false negatives. One last piece of information that I want to show you here on the right is the rare fraction curve, and shows you as you ramp up the species you sample, how many new drugs you get with that have at least one hit. If you do that for antibacterial drugs, the curve saturates very fast. So you found immediately by 5 or 6 species all the drugs that have antibacterial activity. If you do that for human-targeted drugs, actually the curve doesn't saturate. which implies that would you screen more drugs, you would have more of them that would have at least one target. All right. Of course, this is all good. This is all in vitro. So the first thing that everybody wonders is that how do those results translate in vivo? So here I will not show you data, but I want to show you I want to tell you about two things that we did. So actually for the few drugs that there are clinical cohort data there, for PPIs and atypical antipsychotics, what we could do is that we could see a concordance between the species individuals were missing from the microbiome and ones we saw being directly inhibited by the drug in our in vitro screen. But those were very few. So to expand this further and do it for all classes, we went and looked at the side effects those drugs have in humans. And we reasoned that if those side effects resemble the side effects of antibiotics, then this will give us more evidence that those drugs affect also microbes when they're taken by individuals. And this was true. So drugs that have effect in our screen are much more like antibiotic-like side effects than drugs that didn't have effect in our screen. All right, so what kind of therapeutic classes are there? So here I'll show you data. This is all the different drugs that are in the screen grouped according to ATC therapeutic classes, just to make the point that every single one of them has at least one drug that targets gut microbes. Some of them have many more, and here I'm illustrating 3 of them. So calcium channel blockers are used for hypertension, antimetabolites are used for cancer therapy, and antipsychotics. I'm illustrating those to make the point that for antimetabolites, it's quite easy to reason why this is happening, because the targets they have in cancer cells are conserved actually also in bacterial cells. It's much difficult to reason the same way for antipsychotics because bacteria don't carry serotonin or dopamine receptors. As a matter of fact, it's a very vast range of chemistry of antipsychotics. So here I'm showing you on the left a phenothiazine, an indole, and a dibenzooxazepine. Those are different chemical moieties, but they end up targeting the exact same subset of bacterial species. And this, with increasing information about the gut microbiome composition affecting brain function and development, poses the question of whether those effects that the drugs have in the gut might have to do anything with the therapeutic effect they have in humans, and something that we and together with others are trying to follow up at the moment. The last thing that I want to show you from this study is that actually there seems to be an overlap between the resistant mechanisms for antibiotics and non-antibiotics. And I will explain you how we came to this and what are the ramifications. So this is a plot that has all the 40 species that we had in the screen. And each dot is a different species and is color-coded whether it's gram-positive or gram-negative. Disregard the colors is just to show that basically there is no separation that is driven by the general architecture that those bacterial envelopes have. So on the y-axis is the count of how many human-targeted drugs inhibit the growth of this microbe. And on the x-axis, how many antibiotics do the same. And I think you can appreciate that it's a fairly good correlation. And a lot of the species that are resistant to antibiotics, they tend to be also resistant to human-targeted drugs, and vice versa in terms of sensitivity. So this implies that this correlation is driven by common resistant mechanisms. And to test that, we actually took E. coli, which is one of the most resistant ones, knocked out one of the pump that is known to pump out a lot of antibiotics. And if E. coli would become only more sensitive now to antibiotics, then this should move parallel to the x-axis. But what you see is that the mutant of E. coli of the pump actually moves across the diagonal, which means it becomes equally more sensitive both to antibiotics and non-antibiotics. We can do that with another species. This is Bacteroides uniformis. We had a strain that had antibacterial resistance. If antibacterial resistance was only for antibiotics, again, it will move parallel to the x-axis. What happens is it moves across the diagonal. We can go one step further, and for E. coli, we can use high-throughput reverse genetics and assess the role of each gene to the resistance of those human-target drugs. We did it for 6 of them, and what we ended up identifying was the same pumps, transporters, and detoxification mechanisms implicated before for antibiotic resistance. So now, without saying that every single resistant mechanism for any human-target drug will give resistance to antibiotic, what I'm saying is that there's a general trend that the resistant mechanism between antibiotics and antibiotics overlap. Now, this has a certain connotation. And this means that since human target drugs inhibit bacteria, they will select for resistance against them. If this resistance works also for antibiotics, then non-antibiotics, which are taken much more often and for longer period than antibiotics, may be actually a previously unappreciated driver of antimicrobial resistance. And this is something we're currently following up, and we want to map very precisely and understand those cross-resistance and collateral sensitivity interactions between individual drug pairs, as we believe this will open paths for avoiding or even reverting antimicrobial resistance. Now, before I'm going to the second part of the story, I just want to make the point that I showed you that drugs impact, so non-antibiotics impact gut microbes, but I want to make the point also that the opposite is also true. So microbes can also metabolize non-antibiotic drugs. And here I want to show you two pieces of work. One is from Michael Zimmermann, a new colleague here at EMBL, where actually what they did is that they took 270 non-antibiotics and tested them on a panel of gut microbes to see that roughly 2/3 of them, those 176, actually could metabolize directly the drug. As a matter of fact, in many cases, a lot of different microbes could metabolize the same drug. This heat map, the more black it is, the more metabolism there is. They went a step further and they actually identified the enzymes that do that. And they did show also that for a particular drug, this effect has dramatic effects in terms of pharmacokinetics of the drug in an animal model. But microbes can not only biotransform or degrade drugs, they seem also to be able to accumulate drugs intracellularly without that affecting the growth. But although the growth is not affecting with this bioaccumulation effect, this has effects on the microbial community composition. And this is work driven by another colleague of mine, Kiran Patil, and it should also be coming soon publicly available. Now, going to the second part of the story, this has to do with the part that I didn't discuss in the first one, is antibiotics and their effects that they have on the gut microbes. And when you talk about the collateral damage antibiotics have, we take them usually for pathogens, but they do have effects on the gut microbes, it's hard not to mention Martin Blaise. Martin Blaise has been advocating about the collateral damage antibiotics have to our resident microbes for more than a decade now. And apart from writing a fantastic popular science book, which, by the way, if you haven't read, it's a good read, his lab has done also excellent work showing that different antibiotic regimens have tremendous effect on the microbiota composition, and not only that, but they also affect disease development and predisposition in animal models. So this will become important in a minute also. So I will go back to the data I was showing you before, and now we'll focus on the antibiotics, which on a first look, as I went before, looks that most of the antibiotics target all different microbial species across the board. But actually, if you look, there's quite a fine substructure with some of the antibacterial classes like aminoglycosides not targeting anybody, quinolones targeting a generation-dependent effect, newer drugs targeting everybody, older drugs not doing much. Beta-lactams, there seem to be some species that have broad resistance for them, but that has nothing to do with phylogeny, so resistance is strain trait. And then we have protein synthesis inhibitors like macrolides and tetracycline that actually target everybody. So now what I would want to do is that basically pause for a minute and make a subtle but kind of important distinction. So in Martin's work, they mostly use action. So in Martin's work, what they usually use most of the times is beta-lactams and macrolides. And the reason they use them is that those are widely prescribed antibiotics, and they do have broad effects on the gut microbiome. Now, there's a subtle difference between them, and the one that is that beta-lactams are bactericidal drugs. That means that they kill the bacteria they're active against. Now, being bactericidal and knowing that they cannot target everyone, it's easy to reason why this might have a different effect on the microbial composition. So why it might change the microbial composition. It's more difficult to reason the same way for macrolides, which they're textbook bacteriostatic antibiotics. That means they inhibit the growth of the species they target. So now if they inhibit the growth of everybody, then the question comes, how do those have such dramatic effects on the drug microbiome composition? one would expect that when you remove the drug, nobody has been irreversibly removed from the community and the community should resume its composition. So in order to address that, we decided this bacteriostatic-bactericidal divide comes from extensive testing on a very limited set of model pathogens that we have. Those are gut microbes. Most of the people have never worked with them. So we decided we'll have a look again. Before doing that, I have to tell you what is the definition of bactericidal drug. And in order to do that, you have to do time kill curves, which you measure basically survival as a function of time. Now, if there's a 3 orders of magnitude drop in survival within 5 hours after applying the drug, this is what is termed as bactericidal. If not, the drug is termed as bacteriostatic. So we did that for a panel of gut microbes, and we did it both for macrolides and tetracyclines. So macrolides are the blue and the green, and tetracycline is the red. And there are some species that they behave like textbook. So you have those drugs inhibiting them, like Bacteroides fragilis or Fusobacterium nucleatum, but they don't kill them. On the other side of the spectrum, there are species like Rosebura intersticalis, which actually they kill with very fast kinetics. Within 2 hours, you have wiped off the entire population. And there are a lot of intermediate situations where you have slow kinetics, or you have one of the classes killing, but the other one not, like this Purpotella copri here. So you can see the same data just for the 5-hour time point, and broken up according to the different drugs, just to make the point that whichever species falls within this pink area, then this drug is bactericidal for them. And I want to make the point that there is no drug that is either completely bactericidal or completely bacteriostatic. Going one step further is that for some of those species that the drug is bactericidal, like Bacteroides vulgaris and uniformis, which actually are the most abundant species in the gut microbiota, we don't see only that the drug kills them, the drug even lyses them, which is unheard for protein synthesis inhibitors. So this is just a growth curve, and you see the culture actually clearing up, and that's always an indication of lysis, which you can see it much easier in movies. So here what you will see, we put drug to the cells. At some point, the membrane will collapse, and they will dramatically expel the cytoplasmic components out to the media. So what we think is going on here is that it is this exactly selective bactericidal activity that those drugs have that cause the huge effects on the microbiome composition. So they inhibit the growth of everybody, but in some cases, they also kill the cells. And this removes them irreversibly from the community, causing microbiome composition changes. Is there a way to revert that? We reason that there could be if we were able to identify an antidote that would act selectively on a species level. So an antidote that will kill the antibiotic activity for a commensal species, but will still allow the antibiotic to work for the pathogen. And the reason we thought like that is that there was another study from the group showing that when you combine antibiotics across many different bacterial species, the outcome of how those drugs interact is very species specific. So you can have synergies or antagonisms that work for one species but not on the other one. So based on that premise, what we did is that we took tetracycline and macrolides, and we looked for a second drug that would alleviate the effect that they have on bacteroidetes species. So we used the GenderPress-Weak library, and here is the data. So 1,400 different drugs with doxycycline and erythromycin on 2 abundant bacteroides, and we have about 10 hits in each of those situations. We validated all of them. We saw actually that they work on a broad range of antibiotic concentrations, and several of them actually work on a number of other commensal species, which is very advantageous. They also alleviate the killing, and most importantly, they don't affect the antibiotic effect on pathogens. So now having those, I'll show you an example how this works. So this is an example of sulfonamic acid. This is a painkiller. And this, again, one of the complex plots that I'll try to walk you through. So what you see on the y-axis is basically the growth of the different species in the presence of inhibiting erythromycin concentrations. And you see that as a function of increasing amounts of sulfonamic acid. All the black traces are commensal species, and all the purple traces are pathogenic species. So what you see is that when you have only erythromycin, most of the species are completely inhibited. As you increase the amount of sulfonamic acid, a lot of the gut bacteria resume growth, but all the pathogens are still dead. So to sum up this part of the talk, what I showed you is that we have a high resolution map of antibiotic effect on gut microbes. This bacteriocidal-bacteriostatic divide, which is in all over textbook, breaks down completely when one goes outside of a few model organisms. We see that this is a strain-specific trait. We suspect that there are many underlying mechanisms. We're actually trying to find one for E. coli. And I think what was known before we even started the studies, antibiotics have a well-known collateral damage in the gut microbiome. I think what we added here is that there seems to be a dependence on class, and this is also an urgent call for studies that they separate the antibiotic effects and they don't lump them up all together according to their classes. And what we think is happening is that antibiotics with the broadest spectrum and selective microbial killing are actually the most dangerous. And one can discover antidotes that will block the antibiotic effect for gut microbes, which allow the antibiotic to retain activity against pathogens. Now, before I'm close, what I want to say is that what I describe here are all experiments with single bacteria, and I'm sure you're wondering, as we're also wondering, whether those effects translate at all in community settings, because none of us in our gut carries a culture of a single bacterium. So what we want to ask as a question is that do individual species behave the same in the context of the community, or there are also emergent communal behaviors that come up? In order to do that in terms of drug, we actually can probe what is the fitness of every individual in isolation versus in the community, and ask whether this is correlating, as you saw in this plot, or whether you have emergent behaviors. I will not have time to show you data, as this is also an ongoing study, but what I can tell you is that communal behaviors are actually quite common, and they have a lot of underlying mechanisms. More broadly, in the future, what we want to do is that move the complexity of our high-throughput experimental platforms to resemble more what happens in reality, so not single species in cultures, but include also communities, either bottom-up assembled by mixing species, or top-down assembled by using stools, and including the host as part of the culture, or including the host as a readout. By having all these high-throughput experimental in vitro approaches, that gives us tools actually to dissect data-driven observations, validate or refute associations that have been found in data-driven studies, but also generate new hypotheses that then can be tested or design animal model experiments or with the right cohorts. The end game in all this exercise is to generate enough knowledge on the microbial functions and interactions to be able to come with rational strategies to modulate those communities in situ. And with that, thank you for your attention. This is the lab, funders, collaborators who I mentioned through the talk. Thank you for your attention. Happy to take any questions.
Thank you, Nassos. That was a really fascinating talk. As the questions are starting to come in, I have a quick question relating to the first part of your talk. I know that your lab generally focuses on antibiotics, but I'm wondering if you plan to do any similar studies with non-drugs to see how any naturally occurring compounds, maybe dietary or otherwise, might affect the gut microbiome.
Yes, we're actually doing it at the moment. So with food ingredients, also pesticides and pollutants that you have outside in the atmosphere, to actually even excipients that you put into drugs as neutral compounds to see how they affect. I think a lot of times it's quite interesting to see how those interact with the active molecules. So how does an excipient interact with the drug that is usually given. So yes, we're testing that. That being said, what I can tell you, I mean, there's always effects, but I don't think it's a serendipity that drugs are the most bioactive molecules we have. So they do have the most broad effects of anything. Sure,
that makes sense. Let's move on to a question from Meera Krishnan. Meera, are you there? Would you like to ask your question? I will go ahead and read Meera's question. Meera wants to know, can the intake of immunosuppressive medication actually cause a wider range of organisms to then colonize the gut?
As far as I know, I haven't seen studies on that. I mean, what we've done from our side is that we've used immunosuppressants if they have direct effect on microbes, and some of them can actually very well inhibit the growth of gut microbes. Now, of course, within the context of the host, you would expect also other effects. I don't think I can comment on that, or we haven't done those experiments.
Sure. Okay, let's move on to a question from Eric Zhao. Eric, are you there? Can you ask your question?
Yeah.
Go ahead.
Well, you know, the gut microbe where it grows is much more complicated considering the microbe host and the inter or intra-microbe interactions. So I think the culture environment may not adequately mimic the gut environment. So do you think like using the gut organoid or a certain combination of gut microbes will better, you know, optimize or further, you know, benefit the in vitro evaluation of the drug's effect on the microbiota?
Yes, that's very true. So the gut microbiota doesn't look anything like single species growing in rich media in an anaerobic chamber. So this is why we also try to increase the complexity. So go a step further and go to communities that they are assembled alone, or communities of different individuals, including also the host. I do think that all those things will change some of the results, but as a matter of fact, we do see some of this data. There is data on clinical cohorts which microbes individuals are missing when they're taking those drugs. And we actually do see that those are the exact same microbes we see that they're sensitive in an individual screen. So I'm not saying that all results will transpire and when you go to an in vivo setting, but quite a few of them do. And we do see that also with the community setting. And I can say also, it all depends on the perturbation. Yes, so communities have some robustness, but if you can go high enough with the perturbation, then communities seem to behave more like a composite of the individual species.
All right, we have a question now from Prithvanath. Prithvanath, do you want to go ahead and ask your question? Nope, I will read it. So in the case of damage caused by an antibiotic like tetracycline, how is the microbiota repopulated, and will it be the same as it was before the antibiotic treatment?
Well, it's not an easy question. So I guess this reflects to in vivo situation, and I would say it's probably different how it is in animal models versus how it is in humans and different human individuals. There are very few studies actually that have done that in vivo outside of mice. So in a lot of times what happens in the first studies that people have done in humans is that the microbiota regains and comes close, but not exactly to the initial composition. But there are also events that are catastrophic events, that the microbiota goes much further away from its initial composition and never comes back, and also goes to dysbiotic states. But the data that there is there for humans, there is very little. It's not well controlled. Usually there are many different antibiotics at the same time with low numbers. So it's hard to say. For animals, that has been done much more precisely, then a lot of times the effects are irreversible. But I think it's all a matter of dose, and it also is a matter also of the individuals, what microbiota they have to begin with.
And one last question from G2. G2, are you there? Would you like to ask your question?
I think you need to read it.
Yes, I can ask.
Go ahead,
my question. Yeah, sorry. Will the antibiotic antidote that you have used not get transferred to pathogenic bacteria? Wouldn't the trait get transferred to pathogenic bacteria in time?
Could, depends on whether the resistance is de novo resistant or is an immobile element. It could be transferred. And I think this is the kind of danger. The more drugs you use, the more the population size of bacteria that can build resistance increases, and this at some point can be transferred to pathogens. With the antidote, we hope that you remove the antibiotic effect specifically from the gut microbes or abundant gut microbes. So you're actually decreasing the population size that is under pressure to develop resistance. Antibiotic still works on the pathogen, and the pathogen could presumably develop resistance on its own, or get resistance from other things that they're also under pressure and under selection for the same resistance.
Thank you, Nassos.
Wonderful talk. Thank you so much.
Thank you.